Welcome to the second part of the Principles of Immunotherapy segment. In this part, we're going to be talking about how cancer cells evade the immune system. In the first part, we went over the basics of the immune system and the two main arms of the immune system, the innate immune system and the adaptive or acquired immune system. We talked about each of the cell components, how they originate in the bone marrow from stem cells, which replenish themselves and always maintain a stem cell niche, but also divide and create daughter cells of myeloid progenitors for the innate immune system and lymphoid progenitors for the adaptive immune system. And that the innate immune system is a general, non-specific, but fast response as a first line of defense to pathogens and also abnormal cancer cells, our own cells. And the acquired immunity is a slower response because it needs to be learned and adapted, but it is therefore much more specific, and it can also form memory cells that upon reintroduction of the same pathogen or cancer cell, that we can elicit an acquired or adaptive response faster. We talked about each of the cell components, specifically neutrophils and then monocytes, which can differentiate into macrophages or dendritic cells, and how these can do phagocytosis to neutralize and kill cells and pathogens. And also that these are antigen presenting cells that can help to start select and train for what we talked about, T cells and B cells. T cells can be helper cells or cytotoxic effector cells, and the B cells are those that produce antibodies. And we went through some concepts about what an antigen is, what an epitope is, in this lock and key formation of protein-protein interactions. And we also talked about antigen presenting cells, major histocompatibility complex, or MHC, class 1 and 2, also known as human leukocyte antigens, and also cytokines, the molecules that each of these cells use to communicate with each other. So this complex system of immune cells can deal with various sorts of pathogens and also cancer from our own cells. We talked about, if we envision a cancer cell in the stomach or esophagus, that the cancer cells will release antigens as they proliferate and turn over, and that these antigens and damage-associated molecular patterns, or DAMPs, will be brought to regional lymph nodes and uptake into dendritic cells and other antigen-presenting cells that then go on and select for, through specific interactions, MHC class 2 with the T cell receptor on CD4 helper cells, and the MHC class 1 with CD8 effector T cells through their T cell receptors. And that this orchestra of cells, including T cells, B cells, NK cells, as well as macrophages and neutrophils, all effect a response towards the tumor cell in order to eliminate it. We then talked about how this is part of this cancer immunity cycle through 7 steps. First, starting at the area of where the cancer is, and release of these cancer antigens. That antigen presenting cells then endocytose these into the cell and then present them on the MHC class 2 molecules. And that in the lymph node, also known as central training, priming and activation, these dendritic cells and other antigen presenting cells will select for T cells that recognize that specific antigen and activate them and have them proliferate into an army of T cells, all recognizing that same antigen. And then those cells then need to get out into the bloodstream and traffic to the areas of where there is actual cancer and infiltrate into that area, and then recognize the cancer cells being abnormal through that TCR MHC interaction, MHC1 on the cancer cell, and that all of the other checkpoints and other factors in the area have to be conducive to allow for that cell to proceed and kill the cancer cell. So this is what's supposed to happen, and we're going to talk about all the ways that this can break down. So the hallmarks of cancer, all shown here, are all showing properties that a cancer cell has in order to survive. We're going to focus on this major arm here of how cancer cells can evade our immune surveillance and destruction. So with the first step, the release of cancer cell antigens, how can the cell affect that negatively. So we're going to talk about tumor mutation burden and immune editing. So let's start with, again, this cancer immunity cycle where you have cancer cells that have release of tumor antigen. That antigen gets taken in by the antigen presenting cell like dendritic cells in the lymph node, in that central area where T cells can then be recruited, activated, and then proliferate specific to that antigen itself, and then go back to and try and kill cancer cells. But this really depends on the adequate uptake of the cancer antigen, adequate degradation, appropriate translocation to the areas where the MHC molecules reside, and then successful presentation on the receptor, and then successful recruitment of T cells. You have to have a T cell that recognizes that antigen. And it's becoming well known that amongst all the mutations that are present in a cancer cell, very few of them, it's estimated less than 1%, are actually successfully go through this whole step and presented and elicit an immune response. So this is something to keep in mind. That said, when we talk about tumor mutation burden, you'll hear that the higher the tumor mutation burden, the better in terms of eliciting immune response. And the thought there is that if you have more lottery tickets, there's a better chance that you'll win the lottery. But it certainly doesn't necessarily mean that you will have a good response just because you have a high mutation burden. But the thought is that the more mutations, the more chance you have one or more that is an actual immunogenic neoantigen and therefore lead to an immune response. In terms of the tumor mutation burden, which again is talking about how many mutations are present across all the DNA in a cancer cell. What you see on this figure is plotted from left to right, different cancer types based on the highest tumor mutation burden across the cancer to the lowest. And this is called the box plot, and just how to read that. Each of these cancers will have hundreds of patients being assessed to see what their tumor mutation burden is. And what this is telling us is that 50% of the patients reside within this box, and that 100% rely within the outer extremities. And then there's a few outliers that are very rare. And so you can see that the highest mutation burden cancer types are either radiation, ultraviolet radiation associated, or smoking related, the ones with the arrows. And one of those is ours, esophageal squamous cell cancer. But you can also see that the esophageal adenocarcinomas and the stomach adenocarcinomas still rank higher amongst the mutation burden across tumor types. The next thing that you should note is that there is a wide range amongst the stomach cancer patients. And you can see that some have really, really low tumor mutation burden and others have extremely high mutation burden. We see that's common across these three tumor types, stomach cancer, colon cancer, and uterine or endometrial, is that they all have a subset that are MSI high, microsatellite instability high, which we know has a hypermutation profile, as we talked about in the previous video. So they always or usually have a very high tumor mutation burden. On the other hand, we also know that there's heterogeneity among stomach cancer. When we look at the TCGA subsets that we talked about, there's the genomically stable EBV positive and chromosomal instability, which generally have much lower tumor mutation burden compared to MSI high. And that accounts for this wide range of tumor mutation burden across stomach cancer. So we talked about how the higher the mutation burden, the higher the chance that you might have immunogenic antigens from the cancer and therefore a better immune response. And maybe, as we'll talk about in the next segment, a better response to immunotherapies. But we also have to talk about that it's not only the number of mutations, but the quality or the type of mutation. And that gets to going back to these mutations that we talked about, base pair mutations, single variant mutations versus insertions and deletions that create frame shifts and lots of different amino acids. So the single base pair change could create a single different amino acid that then can be incorporated and recognized by a T cell, as we talked about. However, when we look at these frame shift mutations, what we really see is that you can have numerous different amino acids along the peptide chain because of that frame shift, and then an early termination, so the rest of the protein is just truncated and removed. And therefore, you have a much larger segment of neoantigens that therefore have a better chance of being upregulated and presented, and a better chance at having T cell recognition and activation. And so these mutations tend to be more immunogenic than single base pair nucleotides. Therefore, the type of mutation matters. And that's shown here where frameshift mutations, frameshift insertions and deletions are much more immunogenic compared to the single nucleotide variants. And as I was pointing out, although MSI high tumors have a high tumor mutation burden, they also have a lot of frameshift mutations. And so a major reason why these cancers are immunogenic. In contrast, the remainder of the gastroesophageal cancers, chromosomal instability, genomically stable Epstein-Barr virus, they generally have lower tumor mutation burdens, and the tumor mutations that they do have are single nucleotide variants, which are less immunogenic. So again, this explains why, when we talk about in the perioperative setting, why microsatellite instability high tumors tend to have a better prognosis is because they're more immunogenic. And so our immune system is able to recognize them and neutralize them better than the others, especially after removing the primary tumor. Our immune system can clean up the residual microscopic disease that might still be present. In addition to that, there's the layer of the MHC consideration that we have to take into account. And that is, remember that we have a complement of MHC molecules and MHC class 1 molecules and MHC class 2 molecules from our parents that is very divergent from one person to the next. And so, again, in the context of our own personal immune system, are we able to present a specific cancer antigen or not? Someone in their immune system may be able to present a cancer antigen well and create and elicit a response, whereas the next person may not have the right complement of MHC molecules, may not be able to present that on their surface of their cells, and therefore that cancer can evade their specific immune system. So that's something to remember. And then finally, the concept of immune editing. Even if we have a cancer antigen that's appropriately being able to get uptake into the cell of a dendritic cell, be able to go through all the processes, get up onto the MHC class 2 molecule, and elicit a response with T cells, those cancer cells eventually will get eliminated. However, if there's ever a breakdown along the way, and we'll talk about that in a later section as well, that will lead to decreased expression of the antigen in any given cancer cell, those cancer cells will have learned a way to survive and was selected for. And that's sort of the process of natural selection, where the cancer cells that are susceptible to this will be eradicated, whereas those cancer cells that have gained the capability to evade this process will become the predominant problem. And so whenever there's a cancer in place, clearly the cancer cells have found ways to evade the immune system. And one of those ways is to evade this presentation of antigen on MHC molecules. This is by selecting for low tumor mutation burden tumors and also low immunogenic mutations and alterations. In addition to evading this first step of just creating antigens that are less immunogenic or just less antigens altogether, we can also have cancer antigen presentation compromised. How does this happen? Well, the tumor cell itself can decrease its own presentation of antigen through MHC class 1, and we'll talk about that later when we get to within the tumor cell bed. But now, at the level of the lymph node, the tumor cells are secreting many factors and cytokines that get into the lymph node and recruit suppressive cells like tumor-associated macrophages, which are not the good kind of macrophage, or M2 macrophages, T regulatory cells, and myeloid-derived suppressor cells, each of which can secrete more factors that ultimately will negatively affect antigen-presenting cell function by decreasing their MHC class 2 presentation of antigen, so they can't train T cells and select for T cells in the lymph node. And so that's shown here. T regulatory cells can secrete factors and also directly inhibit dendritic cells, so that dendritic cells aren't doing their antigen presentation to T cells, and therefore that step of training and selecting T cells is lost. So in addition then to decreasing the number of antigens or compromising the ability to present the antigens, we can also have checkpoints in the lymph node that prevent antigen presenting cells from selecting, priming, and activating T cells. And that's shown here again. What's supposed to happen is the antigens come into the lymph node. They're presented by dendritic cells through MHC molecules to select for T cells so that they expand, proliferate, and you create an army of these T cells. However, we have checkpoints. We talked about how there's a number of different interactions from the T cell and the antigen presenting cell. And if there is a predominance of inhibitory signal through molecules called checkpoints like CTLA4 and its ligand, PD1 and its ligand, and if we have an overactivation of CTLA4 and other checkpoints, CTLA4 being the really common one within this lymph node at the level of selecting and activating T cells, CTLA4, cytotoxic T lymphocyte associated protein, can downregulate this activation and turn it off. And so again, this is a mechanism that we have to balance our immune system so that we don't get overactive immune systems, yet these should be turned on and off appropriately. And cancer cells, through their various means, they can upregulate T cell CTLA4 expression that then turns this whole selection process off. And this is especially true if and when cancer cells migrate from where they originated into the lymph nodes, where they are directly involved in this with secretion of factors that upregulate CTLA4 expression on T cells and therefore turn off this selection process. And that's shown here where CTLA4 can inactivate the T cell. So in addition now to inhibiting these factors in the step 1, 2, and 3 segments, even if you're able to do all of this properly, the T cells then have to get out and traffic to the tumors and also get into and infiltrate the tumor bed. And so cancer cells have found ways to decrease T cell trafficking to the area where the cancer is and also decrease infiltration into the tumor bed. How do they do that? Well, again, this is what's supposed to happen if the tumor is here, say a lung metastasis, and they are secreting antigens. Dendritic cells are recruiting both CD4 helper cells and CD8 effector cells. They're training them, they're selecting for them, they're proliferating, and now those T cells are getting out in the bloodstream. They need to be able to home to and find where the cancer is, and they need to be able to get into that area. How does a cancer cell avoid that? One is, we talked about how cancer cells need to do angiogenesis. They need to form blood vessels to accommodate the increased cell mass so that they can get nutrients and also get into the bloodstream and start to spread places. But these blood vessels are not normal. They're dysregulated. They don't have the right molecules on the lining cells, which are called endothelial cells. They don't have the molecules that are usually present in order for T cells to home and get into that area. In addition to that, there are cells called cancer-associated fibroblasts that release extracellular matrix molecules, collagen, etc., that form a barrier so that cancer cells can't get into the area well. Cancer cells also secrete factors that shun away the immune effector cells so that they don't register, they don't recognize the area, and they just go right past in the bloodstream and they don't stop there. There are other things that happen within the cancer tumor microenvironment that we're going to get to. So these are all ways that the cancer cell can evade T cells from finding where they are and getting into the tumor area. Even if all of that is working properly, if the T cells are getting into the area, there are ways that the cancer cell can evade recognition by the cytotoxic lymphocytes and other immune cells. We will talk about and have talked about how T cells can downregulate their MHC class 1 expression, and so the cancer cells are not recognized by T cells. So how does that happen? We talked about generally how cancer cells gain the capability of downregulating MHC class 1 as a mechanism to evade the immune system. But in addition to that, we can have mutations in the processing machinery that takes these cancer antigens in the cancer cell and presents them on the MHC class 1 molecules. There are well-recognized mutations in various genes and therefore their proteins that have significant function, including part of the MHC class 1 receptor itself in beta-2 microglobulin being mutated and not functioning, and therefore the MHC class 1 molecule is not working properly. Another one has been in these TAP molecules that help to transfer these neoantigens through the processing. And so if this is not working properly, then the antigen is not being presented on the cell. So in addition to just down-regulating the process, we can get actual mutations in the genes of the process in the cancer cells that then affects the ability to present the antigen on the tumor surface, and that evades a cell kill by the T cells. This is an example of immune editing where those cancer cells that have these mutations would be selected for. They would be able to thrive and survive, whereas the ones that don't have this would be expressing cancer neoantigens and eradicated by our immune system. And so what's left are these cancer cells that have these defective antigen processing and presentation. And so this is the problem. And so we're left with cancer cells that are not being recognized by our immune system. That's again summarized and shown here, where mutations along the way, whether it's an MHC1 itself, like beta microglobulin, or if it's in the TAP proteins that help to transfer and traffic the antigens to where the MHC molecules are. Either way, the consequence is that we're not getting MHC on the surface of the cell, and so the T cells are just not able to recognize it, and the cancer cell is flying under the radar. Now, what about the NK cell, where the NK cell should come in and recognize that this cell now doesn't have MHC class 1, it's not self, and so now it should be the effector cell that then eradicates this cancer cell. So how does the cancer evade both the CD8 T cell by downregulating MHC class 1, but then also evade the NK cell, which should then recognize it as being non-self? Well, that gets to the last step of this cancer immunity cycle, and how cancer cells have many ways to suppress many cells, including T cells and NK cells, in the tumor microenvironment. In addition to that, there is the upregulation of immune checkpoints on the actual cancer cells, which is referred to in the periphery, as opposed to, we talked about checkpoints centrally in the lymph node on antigen presenting cells. So first is this activation of immune checkpoints in the periphery and the tumor cell. And so the way this occurs is that cancer cells, which are expressing the antigens, their neoantigens on MHC class 1 molecules appropriately, can be recognized by T cells. And a consequence of this interaction is that cytotoxic T cells will release interferon gamma. And interferon gamma has the effect of directly acting on tumor cells and upregulating PD-L1 on the tumor cell. And so if there are T cells in the vicinity that are able to kill the cancer cell will upregulate PD-L1, which binds to PD-1 on the cytotoxic lymphocyte, effectively turning it off. And that's shown here, where an active T cell, which is appropriately binding to the tumor cell through its T cell receptor and with the specific antigen. However, when a T cell upregulates PD-L1 expression, which is one of the most common checkpoints, and it binds to PD-1, this will inactivate the effector cell. In addition to that, there are a number of ways in the tumor microenvironment where, similar to as we talked about in the lymph node, now in the area where the actual cancer is across the body, we can recruit and expand regulatory cells like regulatory T cells and the others, which can suppress the function of the T cells and NK cells. We can have metabolic changes in the tumor microenvironment. It's usually very low in oxygen, or something we say hypoxic, and very high acidity or low pH and low nutrients. And so this leads to a dysfunctional T cell and other immune cells. And then finally, the cancer cells are secreting a number of factors, similar to how we talked about in the lymph node when the T cells are being activated. This can then lead to downregulation of immune cells, both T cells and NK cells. So that's shown here in both of these figures, where the cancer cell is appropriately being destroyed by both CD8 cells, T cells, and NK cells. And in the setting of cancer, excreting and creating a tumor microenvironment that recruits suppressive cells, and those suppressive cells directly acting on T cells, NK cells, etc., will effectively turn off the response and lead to an immune escape. So even if all of these processes are functioning, that last process can be the last stand in terms of the cancer cell evading our immune response. So you can see here, going through each of these different steps, that while the cancer immunity cycle should be recognizing cancer, we've seen the various ways and mechanisms that a cancer cell can evade our immune system at each of these steps. The last thing to understand is that these steps can be grouped into three categories. And so the descriptions of each of these three, the first is the immune desert, which is showing that one or more of these first three steps are not working properly. We're not releasing antigens. It's a low tumor mutation burden, or these antigens are not immunogenic, or the dendritic cells are being suppressed and not presenting the antigen in order to select for and proliferate T cells, and or the T cells are being inhibited by checkpoint in the lymph node. So one or more of these will lead to no activated T cells. And so if you go back and take a biopsy of the tumor and you look for whether or not there's activated T cells and immune cells in the tissue, it's completely barren. It's an immune desert. There's no brown staining, which we'll see on the other slides is quite in contrast. So this immune desert is characterized by a paucity of T cells in that vicinity of the tumor. On the other hand, if these are functioning appropriately and adequately, the immune excluded components are when the T cells are getting to the area, but they're not getting into or penetrating into the tumor through the means we talked about, through dysfunctional blood cells, through factors that are being secreted that are shunning the T cells away, through fibrosis and collagen by cancer-associated fibroblasts, so they can't get in. So these are immune excluded. So the immune cells are sort of there waiting, but they just can't get in and penetrate. And then finally, there's an immune inflamed component where if all of this is working properly, step 1 through step 5, but then the T cell is either not recognizing through the mechanisms that we talked about, through downregulation of MHC class 1, or there are an upregulation of checkpoints like PD-L1, you'll have a lot of T cells in the area, but the cancer cells are not dying. And so there's this immunity that's pre-existing, but it's not proceeding, and it's being arrested. And so these specific immune phenotypes are associated with different mechanisms of immune escape from immune surveillance. And so you can see that depending on what the predominant problem is in any given patient's cancer, that the therapies in order to try and stimulate an immune response would be very different. So that's shown here that the immune desert, the immune excluded, and the immune inflamed would likely need different therapeutic approaches to really address the key problems in each of them. And that leads to the third video on cancer immunotherapy approaches. So in this video, we went through the various means how cancers can evade each of the steps within the immunity cycle, and sets the stage for how we can attempt to rescue an immune response at each of those stages. So stay tuned for part 3 when we go into specific cancer immunotherapies. Thank you.